Bangalore. He also got a Professor M.G.K. Menon Lecture Award for pioneering contribution in the field of neuroscience and neurovirology by the National Academy of Science, India on February 28. He was nominated and elected as a Fellow, National Academy of Medical Science 2021. He is nominated and elected as a Council member of Asia Pacific Society for Neuroscience. He is a senior editor of American Society of Neurochemistry. He is an editor of a medical virology virus disease. He is an associate editor of a neuroinfectious disease section, Frontiers in Neurobiology. His specific research areas are molecular approach toward understanding of virus-induced neurodegeneration, basic pathway of neuroinfection and neuroinflammation and viral replication in the brain, human neural stem cell as a model for neurodegenerative disease, cellular differentiation of neurons, glia from human neuronal cells, studies into neuron glial crosstalk for normal and diseased brain function. He has published more than 80 papers in peer-reviewed international journals. 10 book chapters are on his name. He has presented more than 100 international presentations. He got a research funding in crores from various funding agencies like DBT, National Institute of Health, Office of AIDS, research under the Intramural NIH to India program USA. Dr. Seth was first to demonstrate that the severity of HIV-1 dementia is associated with presence of dicysteine motif in HIV-1 TET protein HIV-1 subtype B. For understanding of disease pathogenesis, Dr. Seth developed and perfected a human neural stem cell platform of primary cells of brain. His laboratory has successfully developed protocols for differentiating primary cultures of human astrocytes, neurons, and oligodendrocytes from these human neuronal stem cells. Within India, he made a significant contribution for promotion of a neuroscience with his active role as a General Secretary of IAN. With this great introduction, I would like to welcome and invite Sir to deliver his expertise with our participant on the topic of how viruses affect human brain. Please welcome, Sir.
Thank you, Dr. Rekha, for a really elaborate introduction. I wish my mother-in-law was here, because after 25 years of my marriage, it was a semi-arranged marriage. So yeah, I don't want this talk to be a monologue. So a semi-arranged marriage, which you can understand what it was 25 years ago. My mother-in-law is still not sure whom she was handing over her daughter to. Although the daughter was quite confident because she was a scientist herself. So with that introduction, thank you, ma'am. I will try to live up to the expectations of the audience, which I'm sure is higher now after that introduction. So as you can see, I am coming from the National Brain Research Center, which is an institute which is dedicated to do neuroscience, and it has a mandate to understand both healthy and diseased brain functions. So situated around 40 kilometers away from IGI, which is Delhi Airport. We are situated at the border of Haryana and Rajasthan, and we are still not away from Delhi. And at NBRC, we are trying to understand how the healthy brain works and how diseased brain works. And the idea of understanding the healthy brain is, if we understand how healthy brain works, we can easily understand when diseases happen, that what pathways go back. Now today the topic which I have chosen for this audience is to understand how different viruses cause damage to the brain. Now the first question that should come to our mind is, do viruses get into the brain? Considering that those who have studied biology and brain even in intermediate level, at the 12th level, you know that the brain is protected by something called as a blood-brain barrier. So the blood-brain barrier is supposed to prevent non-needed things to get into the brain. So considering that, that if the blood-brain barrier is there and if it is intact, none of the diseases should get into the brain, and the brain is supposed to be protected. But even then, sometimes the viruses and pathogens make their way into the brain. So our lab is trying to understand when such things happen, when infections reach the brain, how do they affect the brain cells and brain functions. So the three viruses which we are interested to understand are HIV, which we all know causes AIDS, Zika virus, I'm sure some of you would have heard this name, which causes something called as microcephaly. And the name itself indicates micro is small and cephaly is head. So small head size. So if the infants are born to mothers which are infected with Zika virus, and if they have abnormally small head size, we want to understand why that happens. And then of course the SARS-CoV-2. This was more of a forced research when everybody was getting into the SARS research. There was a big meeting at Department of Biotechnology, and Brain Research Center was said, आपका कोई role नहीं है, आप सिर्फ SARS testing कर दीजिएगा हमारे लिए because you have PCR machines. But I was trying to tell to my director, sir, and the reason nobody wanted to believe, because there was enough evidence to understand that it is a lung infection. What does it have to do with the brain? But if you remember, if we have heart attack, there's a short supply of oxygen into the brain. Same oxygen is coming from the lungs. So I was of the opinion that if lungs are affected and oxygen is going down, there's bound to have a damage into the brain. And we wanted to understand. But I was kind of shooed away that time. But later on what happened, we wrote a review. So when the labs were closed, we had nothing else to do. So me and my students said, sir, ek baith ke review likhte hain. So I said, I know the topic on which we'll write. And the reason to believe why SARS-CoV-2 can also have an effect of brain was coming from the initial evidence that in SARS, not CoV-2, but SARS alone and MERS, the two other diseases which were parents of the SARS-CoV-2, there were evidence to suggest that the virus makes its way into the brain. So if any pathogen gets into the brain, it is bound to have an effect. And unless we understand what effect it causes, the research will not make way. And we can't treat the disease. We can't understand the disease, forget about treating the disease. So with that intent, we got into the SARS-CoV-2 research, and later on it was proven by majority of evidence that it has a component of brain fog, or something what we call as post-sequelae of SARS-CoV. So I'm not going to talk to this on this topic today, because if I talk about everything, maybe Madam will not invite me. And thanks to Dr. Preeti Sharma, who had extended that invitation. So I'm saving this brain fog topic for next time, hopefully with it. Now, so please give me a good review if you have a feedback, so that they will invite me again. So coming back to these two viruses, and the reason we have chosen these three viruses are because, not only because they cause damage to the brain and cause these diseases, which is called as HIV-associated neurocognitive disorders. So HIV being an immune disease which brings down the immune cells in the body, and then we fall prey to the opportunistic infections, and that is why people die. So the people die because the immune system comes down. So we know HIV/AIDS is an immune compromised disease, but it also causes damage to the brain cells, and it causes something called as HIV-associated neurocognitive disorders, which is also briefed as HAND. Okay, so as the name itself is indicating that this virus causes something in the cognitive function, and that decreases the neurocognitive ability of these individuals, the AIDS patients, and that is why it is important to understand its role in HIV in brain. So whenever you want to research on something, you need to have need a model system. If we are working on plants, we need a plant system. If we are working on brain, we need something as a holistic organ, a brain, or an organism which has a brain. Most of them have. So accordingly, we were supposed to choose a model system. When I started my work in HIV after coming back from NIH, HIV is a human-specific virus. In other words, if you take HIV, put it in any animal, nothing will happen. Accidentally, during your experimentation, you poke your finger, there's a chance that you will contract AIDS. So we needed a human system. So animal system was not there. The choice of animal system was not there. And then we wanted to have brain origin cells. And then thirdly, we wanted to have a system which is least tampered with, and it is as close as to the normal physiology. So for that reason, we started with developing something called as human fetal brain-derived neural stem cells. Okay? Now why stem cells? I'm sure all of you know that stem cells have the ability to make any cell type. Like when a baby is born, depending on what kind of nurture and nature we provide, that person can become a doctor, engineer, a dancer, a terrorist, or whatever. So depending on what cues you give to that individual, that will differentiate or develop into that. Similarly, neural stem cells have the ability to form either astrocytes, neurons, oligodendrocytes, and other cell type of the brain. So with that ability in mind, we wanted to develop a model system which will be good enough to understand the disorders of brain, and that is why we started with a model system called as human fetal brain-derived neural stem cells. Now, as again the name is indicating, human fetal brain-derived, so the brains which we are using from fetal brain, where do we get them from? We get them from elective abortions due to family planning reasons or whatever other reasons. We follow all ICMR ethical guidelines and stem cell guidelines. We get the fetal tissue, bring them to the lab, open up the brain, get into the areas which are rich in stem cells. And if you also notice, the gestation week is 8 to 16 weeks. So usually there's a 40-week gestation, 9 months. And here we are trying to target 8 to 16 weeks. The reason for that is the time when you have the maximum number of stem cells, because the brain is not developed into, and that is also the time when the legal abortions are allowed. So that is. Now I said that all in a very simplistic way, but it took me 2 years to develop this model system. 1 year to get the clearances, and the second year to develop the model system. And once we have these brains, we basically culture them in different ways, 8 to 7 days, 5 to 7 days, with and without serum media. Cell passage them. Those who do cell passaging know that, if you split the cells, put it in another flask, that's one passage. So you passage them multiple times. And then depending upon the morphology and cell-specific markers, you can identify that the population of cells which you have in your plastic plate or Petri plate is actually stem cells or not. Now how do they look? So this is portion of a brain which I have tried to photograph. And that time, I'm talking about 2003 when I had joined NBRC. There were no sophisticated microscopes at all. This is taken from a Sony camera, वो जो क्या था, Sony handy cam कुछ करके आता था, उसका objective पे रख के these pictures were taken. And if you see these pictures, they show you a piece of brain, fetal brain, which we have taken from day 1 to day 8, and there next slide you will see it up to day forward many other days. You see that the brain sprouts out some cells, and as the day passes, it's a picture of the same brain portion, and you'll see the number of cells which are sprouting out from this brain portion are immense, and they just fill up the flask. They grow as weeds once they start dividing. And if I shift this, again you'll see they are growing as a beautiful monolayer of cells, and they're dividing so fast because in just day 8, they fill the entire flask. And they're dividing so fast because they have the ability of self-renewal and high proliferation rate, and that is why they are stem cells. So one of the thing which I was very sure with me that they're stem cells is because of their high proliferative rate, but that's not enough. Now stem cells, as I said, are unspecialized cells. And by unspecialized cells, I mean they are not astrocytes, they are not neurons at that time, but they are only cells which can give rise to all these cells. Now how do I know? Can I get the lights low, at least in the front if possible? So you will see that we do some certain cell-specific marker, and the procedure which we use is immunostaining, where you take antibodies against certain antigens. And since neural stem cells have this antigen called nestin, which is a marker for neural stem cells, when we stain for them, we see that the majority of the cells in this frame of culture are all red, which are basically means that they are positive for nestin, which shows that they are neural stem cells. But at that time, their other markers which we tried was GFAP and TUJ1. This is a astrocytic marker, this is a neuronal marker, and none of the green cells are there, which also tells me that they are pure population of neural stem cells devoid of any astrocytes and neurons. The other important property of these neural stem cells is to form a neurosphere. Neurosphere. All words are telling you what they mean. So sphere coming out from neural stem cells are neurosphere. The other cell types which have this property of forming spheres is the cancer stem cells. So when we tried to make neurospheres out of them, they also were made. And then third confirmation that whether we had the neural stem cells or not was when we gave them the proper media conditions, we could make astrocytes or neurons from them. I think my charging was not on. चार्जर ऑन नहीं किया था ये स्विच आ गया ओके ये तो यहाँ से ऑन करना पड़ेगा ना चार्जिंग ऑफ थी 1.5-2 घंटा लगा था ना आ जाएगा just bear with me for a minute. So while this is booting up, so we figured out that they can make spheres, and if I give them conditions for astrocytic media and neuronal media, they would make either astrocytes or neurons from them. Now once we were sure that they are making these specialized cells and they are themselves unspecialized, we were sure that they were neural stem cells. All right.
Use this as a pointer. ये भी चलेगा.
So I think it's booting up, so it's kind of frozen right now, but let me tell you what I'm showing in this slide. I hope it doesn't move from here. So what you're seeing here, we had a neurosphere which we started differentiating into stem and into neurons. So we started following a process of neurogenesis, which is genesis of new neurons is called neurogenesis, which is the most important thing. If we can make adult brains do a lot of neurogenesis, then most of the neurodegenerative processes can be prevented or repaired. So what we are trying to do here is to make human neurons in the Petri plate. So all we do is take neural stem cells which are all nestin positive, negative for TUJ1, neuronal marker. We push them into neuronal media, and what you see after 7 days is that you see some green cells which are indicating that new neurons are formed. Then we are seeing some yellow and orange cells, which means they are into differentiation. Why they are yellow and orange? Because they are staining both for the nestin marker, which was in red, and green in neuronal marker, and that is why these are cells which are undergoing differentiation. Okay. All right. So now the fact that they form neurogenesis or they can form the neurospheres is indicated in this time-lapse microscopy. So we have these microscopes which can go and take pictures at you fix the time, and then they can make a movie out of it. This is how you do time-lapse microscopy. And you're seeing one neural stem cells dividing into 2 and then 4, 8, 16, 32 and so on, and it will form a sphere-like structure. Now this assay system gives us an ability to understand the proliferation rate. So I can fix the number of cells to start with, and then at the end of one day, 2 day, 3 day, I can measure the number of cells. I can also measure the diameter of this sphere which is formed using the softwares, and then I can understand the proliferation rate of this. Now why do I need this assay system? I need this assay system to understand if I have some viral proteins or pathogens or some plant products which are good or bad for stem cells, good or bad for making new neurons. I can put them into this and then do the assay. So if I have a control slide, if I have another set of things, and I can repeat this assay, I will figure out by measuring the diameter whether it increases or decreases the proliferation rate. And I'm going to tell you how we use this later on. So as I said, we use this model system of neurospheres, which is suspended culture. This is a much more advanced 3D culture system, and also as a monolayer. And we have tested them for a lot of markers such as nestin, SOX2, Ki67 and so far. And using this model system, we have published several papers which basically indicates that this model system is accepted worldwide. And you'll be proud to know that we are Atmanirbhar on neural stem cells, and we are the only laboratory in the country and very few in the world which have human fetal brain derived neural stem cells, which are primary cultures, and they're working fine with us. So this stem cells we also give out as a mandate for empowering the neuroscience community in the country. Now, what is the potential of this immune system, the model system? It is immense. And it is immense because we have used to study how HIV affects the stemness of neural stem cells. We have also used to understand how HIV affects the neurogenesis. Again, we use those model systems. We've studied the effect of HIV on astrocytes, neuron glia mediated, Zika virus on human neural stem cells, and SARS-CoV-2 neuropathogenesis. Now, this model system provides us immense potential for opening neuroscience research in the country, and we have been very successful in doing that. So once we were very confident with single cell culture system, either astrocytes, neurons or neural stem cells, we started to venture out into more difficult problems, where we started to having co-culture system. In brain, neurons are never there only in isolation. They are always together with astrocytes. In fact, each neuron is surrounded by 50 to 100 astrocytes. In fact, without astrocytes, the neurons cannot function. Now, knowing that, it was important that we establish a co-culture system. We have double the number of astrocytes, half the number of neurons, and try to grow them in co-culture. And we did that. And what we find is something like this. Now, again, stain for neuronal marker is green. All neurons are stained green in this presentation. So this is neuron over here. And as you can see, it will extend its processes to look for red cells, which are astrocytes. So when they find these new astrocytes, they will go and stop there. And these processes are going and extending their processes up to the astrocytes. And why are they doing that? Because they get a lot of good things from the astrocytes, and they dump off all their waste onto the astrocytes, and then astrocytes do the housekeeping function. So for this normal physiological function, it is important. The brain is dependent upon this crosstalk between the interplay between the astrocytes and neurons, and that is why this model system was developed. So this model system basically is a very robust model system which helps you to understand how neurons function in the presence and absence of astrocytes, and if something happens to astrocyte, what will happen to neurons. So all neuronal function are dependent on astrocytes, can be studied by that. Now, one such example for that is we used a virotoxin called as TAT. TAT is transactivator of transcription, a protein which HIV virus uses for its own replication. So when any pathogen gets into a host, the first thing it wants to do is multiply. This TAT protein is very, very important for multiplication of HIV. But unfortunately, this protein is also very bad for neurons. So it's a neurotoxic protein. So this neurotoxic protein, if I put it in this culture system, the first thing which I see, these processes of neurons which were formed, within 24 hours they start retracting them and they give this bead-like appearance. This is the first step before they die. If I wash off the TAT from there, from the culture media, these processes go back and attach to the astrocytes. If I leave the TAT there, all the neurons will die. Now this is a way I'm trying to tell you how neurons can be studied, effect of viral proteins can be studied. Now this picture, does it give you any idea what I'm trying to talk to you about? Now this is a very famous story. There was a fort which was almost impregnable, and two great emperors were fighting. The one outside the fort almost gave up, but came up with an idea that I will present a horse, a wooden horse, to the person as I gave up, I can't win you, I want to be a friend, I want to present you something. So he proposed to give a horse which was unfortunately laden with soldiers, the enemies. And the other fort, they lowered their gate, allowed the horse inside, and that helped the army to get in. That's exactly the mechanism which HIV and some other viruses are now found to be using, and that's called as Trojan horse hypothesis. Okay, that's the way, or the only way, the most common way I would say, for at least for HIV, the way by which the virus gets into the brain. So let's try to see in this animation how it gets into the brain. Now you're looking at a cross section of HIV virus. Now, irrespective of the route of infection, whether it is blood transfusion or risk receptor behavior, you will have the virus come into the brain through this blood. Let me try to see if I can stop this. Yeah. So this is what you're seeing is a blood microvasculature in brain. Brain is very rich in blood microvasculature. Here you're seeing a cross section of a microvasculature where you're seeing an RBC, you're seeing a blue cell which is a monocyte, which is a blood cell type, and you're seeing also tight junctions of these endothelial cells. So the brain, as I said in the beginning of my talk, is protected by something called as a selectively permeable barrier, which is known as blood-brain barrier. It's a barrier between the blood and the brain, which prevents or selectively allows few things from the blood into the brain, and only those things in the brain which are important for the brain or needed from the brain. Something similar to the security guard in outside our community or outside this university who checks whether this person is okay to get in or not. That's exactly what the blood-brain barrier models provide. Now, it is also not selective based on the size of the cell. So something which is as small as virus, which is in microns, that cell, that can actually get into, that cannot pass the blood-brain barrier, but something as big as monocyte can pass through the blood barrier, because the selectively permeable barrier allows the monocytes to pass through the blood-brain barrier. So let's see in the rest of the animation. You're seeing these white particles are HIV virus particles, and you're seeing a monocyte which is not infected, and here is an infected monocyte which can pass through the blood-brain barrier, and this is how it fools or breaches the barrier and gets into the brain, gives the viral particles out. It looks for a CD4 receptor, and you see the nucleic acid getting into the brain. So what you saw in this animation is trafficking of the virus into the brain. Okay, and this is called as Trojan horse hypothesis. Once the virus gets into the brain, the problem starts. And what you saw in the animation was this part, okay, breaching the blood-brain barrier. Once it gets into the brain, now you see the HIV virus which is now in the brain. It gets into the brain, does two things. Number one, it sheds out its proteins. So these proteins which are there as a spike protein, let's say, for not spike exactly, but they look like spike of HIV are GB120, which is glycoprotein 120, that protein, and the TAT protein, which I said, which is important for the multiplication of the virus. These two proteins are the most neurotoxic protein which are given away by the virus in the process of its multiplication. And those two proteins are bad for the neurons. Now the other interesting part is that the neurons are never infected, but they are affected. Now why neurons are not infected? Because number one, they do not have the CD4 receptor by which the HIV can go and dock to that. Second, neurons are a terminally differentiated cells, so most of the viruses and pathogens do not infect cells which are terminally differentiated, because viruses are dependent on the host cell. If the host cell is also kind of not active and dividing, they are not interested in infecting them. So they go and hide and they infect microglial cells, which is another cell which is present in the brain, microglial cells as well as astrocytes. Now if you remember my introduction of neuron and astrocyte co-culture, I told you if astrocytes are sick, there will be a problem for neurons. Now what do astrocytes do? Astrocytes, as I said, provide all the good trophic factors, oxygen, glucose to the neurons, and they take away the excess of neurotransmitter glutamate, which is built up at the synaptic site. So when there is synaptic activity, neurotransmitters are released from the presynapse on the postsynaptic neuron. Only 20% of that neurotransmitter is used for synaptic transmission, 80% is extra. Now why should a cell have an extra neurotransmitter? Is because to make that process very efficient. In other words, neurotransmitter should not be limiting, that is why it makes. because it knows that the extra 80% is going to be absorbed by the astrocytes, which are there around the synapse. So they take that extra neurotransmitter, recycle it and put it back in the neurotransmitter. So this is a normal physiological function if astrocytes are functioning fine. So most of the things, I know the simile which I gave of this part of role of astrocyte is of the lady of the house. If you have your mom, your maid or somebody who's doing all the chores but we don't recognize that person, but if that person falls sick or stops doing their job for a day or two, maybe first day you can manage, second day maybe manage, third day it will be problem. That's exactly what happens in astrocytes. Now what happens in astrocytes, if they stop taking the glutamate, that extra neurotransmitter causes excitotoxicity and it kills the same neurons for which it was important. So that excitotoxicity, death by excitotoxicity of the neurons is the way by which neurons are being affected. So they are not infected but are affected because the cells which are providing the support to the neurons die or they get sick. They don't perform their functions well. That's exactly what happens. So there's a direct cell death by the neurotoxic proteins or the indirect cell death because the astrocytes stop performing their protective functions. So either of the way, what happens is neurons die, and that is when the problem of neurocognitive deficits start. So the very first question that we started to ask with that model system, and I'm not going to give you data, I'll just give you conclusion slides, so please bear with me. I'm surprised that after the post-parental and sumptuous lunch, all of you are awake. I was thinking I will go to sleep, but my audience will definitely go to sleep before, but I'm really, really thankful to you that all of you are awake. I can't see, even at the end, last person is awake, which is very good. Now, so hang on with me. So, when there was a time in 2000, early 2000, when there was a belief that India was number one in HIV cases. In the West, there were a lot of neurocognitive deficits which are being reported. The hand problem, okay? Now, it was very obvious that why hand was happening is because neurons are dying some way. But how they were dying was not known. That is where our lab and several other investigators from around the world have helped to understand how hand occurs. But the other question was, why in Europe and US there was a lot of hand and in India there was less hand. But the number of infections of HIV were more in India as compared to US and Europe. And the question was why there is low incidence of HIV associated dementia in India. And this was the question which I took up as a challenge to start my lab and start working on it. Now this was a high risk, high reward project. High risk because nobody wanted to do this, because nobody knew how to do it. High reward because if we could make a scientific basis as to why there's low dementia in India, then probably the rest of the world can get over it. So as was shown, as was told by Dr. Rekha in my introduction, that we found out for the first time that the subtype TAT-C, the TAT protein from the Indian strain or the subtype which was affecting the India population was HIV-1C. जैसे आपने COVID में सुना ना Delta, Omega, Omicron. So in those days, this clade concept was not that much, but there was a clear cases that entire 95% of infection in India and China were all of HIV-1 subtype C. Now, if I take this subtype B virus and subtype C virus, and I take out the TAT protein of both the virus and then do a proteomics on them and try to figure out, there's a mutation, a natural mutation which is there in subtype C, which is not there in subtype any subtype of HIV actually. HIV comes in different flavors, A, D, E, K, C, B. But in C only there was one mutation which was unique to C. So we tried to harper on that and we figured out that if there's a dicysteine motif mutation, it causes less neurotoxicity. And the reason we showed it that was using the same model system. We took the neurons from human, we put them in TAC-B protein, TAC-C protein and figured out that TAC-C protein is less toxic. And this is how we made this major discovery. And we have this highly cited paper which gave us huge visibility to the institute as well as to us. And this laid basis for a lot of foreign grants as well as collaborations with Johns Hopkins as well as NIH. And this was the first student who's now actually in WAPI in one of the companies here. I think she's at the J Research Foundation. She's a senior scientist. Mamta Mishra. She was my first research student who took the challenge of establishing the model system with me. Those were the days when I was also working in the lab, and we proved that this is why we have less hand in India. And then we just started. Once we were confident and we got all the support and attention of everybody, we just exploited the model system which we have. So the biggest problem initially as you step into research would be first to identify a research question, and second is to develop a model system. Once you have these two, then the sky is the limit. So that's exactly happened with us, and we started asking series of research questions. The second question which we asked was, how does viral proteins affect the proliferation of neurostem cells? Do they do anything to stem cells? Because once we knew that the virus was getting into the brain, we wanted to understand what the virus does to all cell types of the brain. So we showed in neurons, astrocytes, and then we looked into the proliferation of these neural stem cells. And here what we found was cyclin D1, which is another cell cycle regulator. It's important for cell cycle, if you have studied the cell cycle. And we found that when we put the TAG protein there, the cell cycle is disrupted. As you can see over here, without TAG and with TAG. All the green cells are gone in with TAG, which basically just tells you that the proliferation of this was prevented. Once we figured out that the proliferation is prevented, we wanted to get deeper into the mechanisms, because if you want to do a good research, you have to move from observation to mechanism. And that's what we did. And we published another paper where we showed that something called as tripartite motif 32, which is in abbreviation is called as TRIM32, modulates the proliferation of neurostem cells in HIV. And how we showed was, because we had this question that what causes the quiescence of neural stem cells, the presence of TAG? Is it mediated to TIM32? So all we had to do is either overexpress this TIM32 in our neural stem cells or block the TIM32. And we did exactly that. We did both manipulations, and we found out that TIM32 is important for bringing the proliferation rate down because of TAG. Now, everything what we did in Petri plates, although it was human cells, primary cells, they were not cell lines, they were primary cultures, we always validated them in patient samples. So there's something called as a human brain bank, which is again unique in the country. It is there in NIMHANS, Bangalore, which is National Institute of Mental Health and Neurosciences in Bangalore. They have a human brain tissue repository, HBTR, and they happily give you sections both for teaching and for research. And they have a huge collection of brain samples of different disorders as well as infectious diseases. They also have sections of COVID-19 brain samples. So we requested them to provide us brain samples so that we can actually relate our Petri dish research to the human population. And we found that indeed there was TRIM32 was very highly expressed in HIV brain patients. Okay, so that was a proof of the concept. And then we published another paper to show that how TRIM32 is modulating the neuropathogenesis. We found out that one microRNA is important. I'm not going to go into these details, but we'll be happy to discuss if anybody is interested. And this is how our research went forward. And the credit for that research goes to Meher Fatima, who is at now St. Louis University. Now, this is another microRNA which we found out in HIV patients, which was relevant. And this is how we found this whole hypothesis that how HIV is causing damage to the brain, both at the neural stem cell level and at the astrocyte and neuron level. And then it was, as I said, there were a lot of parallel papers which were coming up. This work came out of the thesis of Manju Tiwari, who actually figured out that the damage to the neuron, as I said, is mostly through the astrocytes. She figured out that what receptors, what pathways are actually causing damage to the neuron. And then Chitra continued this work, and she figured out that something called as epherin, which is important for the crosstalk between the astrocytes and neurons, is also affected, and that causes damage to the brain cells. And then when we were doing HIV, and this HIV was our first club, it continues to be there, first clubs you never forget. So HIV continues to be a part of our research, but then there were a lot of outbreaks of Zika virus. And very quickly, just bear with me, there are just a few more slides left, and I will breeze through them. And this virus came into news. The reason why it came into news was because it coincided with the Brazilian Olympics, where there were a lot of outbreaks of the Zika virus, and simultaneously there were news of a lot of kids which were born with abnormally small head size, right? So there was some correlation that there's maybe this, because of this outbreak, this Zika virus is causing microcephaly. Now what is the Zika virus? It's a flavivirus. It is carried by the Aedes mosquito, the same mosquito which carries malaria, chikungunya, dengue. So all the mosquito has to do is load one more virus to it, and just one bite is good enough to impregnate you with all these viruses. And the damage, if you take the structure of the virus, all viruses are made of a structural protein and non-structural protein. So the structural proteins are those which mean the structure, and the other non-structural proteins. So what we did was, we took the structural as well as the non-structural proteins. Now the other thing what we have always done in our lab was, we never went for the low-hanging fruits. Now why low-hanging fruits we don't go? Because we lose the race. What happens by the time we start reading, somebody has already shown that Zika virus causes damage to the neural stem cells. But then people leave it there, because the most challenging work is to figure out how does Zika virus cause damage to the neural stem cells. So we pick up from there. So we took structural proteins and non-structural proteins, took all the proteins overexpressed in different petroplates of neural stem cells, and started looking at the proliferation. What assay system we used? We used the same Neurosphere assay which I showed you in the time-lapse microscopy. So we found that when you put neural stem cells, you look at the proliferation rate, you look at the death rate, you look at the damage or properties of the neural stem cell. What we found out of all that research was that the E protein, the envelope protein of the virus, causes the maximum damage to the proliferation, and then we started the research there. So this is what I said. And the other interesting part is that Zika virus causes microcephaly up to 7 to 10% of the infants which are born to the mothers which are infected in their first trimester. If the same mosquito bites in the second or third trimester, chances of microcephaly are very, very low. So what does that tell you? That tells you that there's something special happening in the first trimester. Okay? So people were advised not to go into countries which are rich in mosquitoes or Zika virus or conceive in countries during that time, because they were highly likely to have microcephaly. In the first trimester is the time when the maximum development of your head and brain occurs. If you remember, those in the family, when a woman is pregnant, there's a repeated ultrasound at least 3 times during their pregnancies for measuring the head circumference and other growth parameters. So that first trimester is important for the development of growth, and that is why if the Zika virus infections occurs during first trimester, there is highly likely that you will have microcephaly. Now how does that happen? Was the question. The other interesting part is, while HIV crosses the blood-brain barrier, it never crosses the placental barrier. Zika virus is notorious that way. It has the ability to get into the inside of the fetus during pregnancy in utero, which means it has the ability to cross the placental barrier. It also causes the blood-brain barrier, which we showed later, but it also has the ability to cross the blood-brain barrier, the placental barrier. And that is why it causes, this is a normal head size and this is an abnormal one. Is this real? Does it really happen? Yes, it does. And you can see this is the outcome of the microcephaly. Now what is the future of these kids? Very, very bad. Most of them die in utero. Stillbirths are happening. Then later if they survive maximum for 10 to 12 years. But those 12 years are horrendous for the patient, for the person as well as the caregivers. So obviously this is a bad thing, right? Even if Zika virus does not kill people, it can have a bad consequence. And that is why it was a question of research. And as I said, we started looking at structured and non-structured proteins. And what we found was the envelope protein actually disturbs, if you put it, overexpress them in neural stem cells and then do RNA sequencing and sequencing, you will find that the microRNA circuitry is disturbed. And we picked up the microRNA which were overexpressed, downregulated, and are researching on that. And we have found that 2 microRNAs, 2043P and 1273, which affect the notch and PAX, which are important for the stemness of the neural stem cells, they are affected. They affect the process of neurogenesis, and they result in a smaller sphere size. So all these three things told us that they are affecting and causing, why they are causing neuropathogenesis. Reshma delved deeper into it and she figured out again different other microRNAs which were affecting, and by various pathways we have figured out how Zika virus envelope protein is bad. If you look at this neurogenesis happening from the neurostem cells, the same brainstem, the neurosphere model, you take a neurosphere, put it in neuronal media, it starts giving new neurons. The new neurons are good if they are branched and they migrate well. In the presence of E protein, that migration and branching is stunted, which means that their migration is less, and that means they are bad neurons, and that is why the development of the brain doesn't occur. Okay, and then again, we are continuing this work, and Himali Arora, who just completed her PhD and now she is moving to UIC, University of Chicago, for a postdoc, showed that there are certain long non-coding RNAs which are also being affected. And then again, when we were confident with Zika virus research, we just got more adventurous and we started to form the blood-brain barrier model. Now again, the blood-brain barrier model, as I told you in that animation, it is comprised of brain endothelial cells which are tightly packed together, and they are supported by the astrocytes at the joints. So we formed a blood-brain barrier model. Again, this is again unique, and only we in the country have this model system, which is made up of human cells, primary cells, and 2 cell types. And how do we form them? We form them using this transwell apparatus. So this is one well which is hanging in a 24 well plate. You place, you fill it with media, you place 1 cell type, endothelial cells. So this is your blood side, this is your brain side, and here you will have to put astrocytes to make it a complete blood-brain barrier. Now if I put my dye over here, it should usually not pass if my barrier is intact. But if I disturb this barrier, my dye will come and can be passed through this. So these are few tests which we use. And using this blood-brain modular system, we showed that Zika virus can cause alteration in the blood-brain barrier model also. Okay, so the E protein again disturbs the brain microvascular endothelial cells and the astrocyte layer, and which causes the disruption in the blood-brain barrier. So what is the conclusion? That's my last slide, is that we have developed several lines of human fetal brain derived neuroprecursor cells or neurostem cells in the lab, which is a unique research neuroscience resource for the neuroscience community in India. TAT causes toxicity, causes damage to the brain cells, which causes neurocognitive disorders. TAT also affects the neuron rehab crosstalk. Zika virus disturbs the microRNA circuitry. We have formed the 2-dimensional contact-based human blood-brain barrier model, which we are using now, and Zika virus protein causes damage to the tight junctions as well as causes pathogenesis. Now, over the period of these 20 years, I have been there at NBRC, National Brain Research Center. Out of the need of the research, this model system developed. Okay, and that is culture of primary cells, neurosphere model, co-culture model system, and there were some times when we were having difficulty in getting the fetal tissue. Okay, this happened when the lot of over-the-counter abortion drugs were available and people didn't go for abortions to the hospital. Our source for the fetal tissue was from the hospitals. So fortunately, because we have a huge pool of these neural stem cells which we can cryopreserve and bring them back to life out of liquid nitrogen tank, although they are primary cells, we are not dependent on a regular supply of fetal tissue. But at that time we started making iPSCs, which is induced pluripotent stem cells, which we developed from human blood. So if you give me 1 ml of your blood, I can give you your neurons. That is a promise, which basically is a model system, again a model system which we can use, and we have established several collaborations out of this platform. And that brings me to the credit slide. We are a lab of very happy people. I think the happiness index of Surat is much higher than Gurgaon, because we are very stressed out otherwise there. You see everybody smiling over here, but usually we don't, right? So whenever we get a chance, whether it's acceptance of a paper, got a grant, somebody graduating, somebody's birthday, I've told them just don't celebrate my birthday because that reminds me I'm getting old. So we celebrate only decade and half decade. मतलब जब मैं 40, 45, 50, 55 आगे नहीं बताऊंगा अभी कितना है. So this is, I think, one of the parties when Reshma was coming back. So she is a postdoc outside. So she was visiting us. So we went to a high-end Hawaii in Gurgaon. And this is a picture. And these are other people who are, you know, not with us now. They're ambassadors of NBRC and doing well. So this is my lab. Not all people are here, but many are mentioned over here. And then we are heavily dependent on civil hospital, collaborations with civil hospital. NIMHANS Bangalore for brain sections from Dr. Anita Mahadevan. Research, some good leads have come out from our collaborations from Abindranath and the director of the clinical director of NINDS and Dr. David Wang, who is the translation director of translation research there. And nothing goes without money. Money comes from different sources which are listed over here. And thank you so much for your patience and for staying awake. I hope I have not crossed the time limit. Thank you so much. I'll be happy to answer any questions. Yeah.